The protozoan, Lophomonas, has been reported to be a pathogen of humans in a series of case reports. Most of these case reports describe infections of the respiratory tract. Today we will talk with two experts about this possible parasite. Some of the questions we will address include: what are the characteristics of the species in the genus Lophomonas? What are the laboratory and clinical data that suggest that Lophomonas species cause human infections? And finally, is Lophomonas truly a pathogen of humans? Welcome to Editors in Conversation. Please subscribe to this podcast and rate or review it. This episode is brought to you by the Journal of Clinical Microbiology, available at jcm.asm.org. I'm your co-host and JCM Editor-in-Chief, Alex McAdam. Editors in Conversation is supported by the American Society for Microbiology, which publishes JCM. If you are a member of ASM, you can get up to 50% off the publication fees when you publish in JCM or any of the ASM journals. I am joined, as always, by Dr. Ellie Thiel, co-host for the JCM episodes. Ellie is an editor of JCM and a clinical microbiologist at the Mayo Clinic. How are you, Ellie?
I'm pretty good, Alex. How are you?
I am fine. We got kind of an exciting invitation, you and I, this week.
We did. Do we tell people about it, or do we just leave it there?
Well, I think we could mention the session that we've been invited to participate in, but we're still kind of hammering out what we will be doing in the session. But we will be doing something in the Battle of the Brain session at ASM's upcoming microbe meeting.
That's right. I'm pretty excited. It's going to be really fun.
Are you a regular attendee?
I am. It's one of my favorite sessions. I think it's one of everybody's favorite sessions. It's so exciting. You get to see kind of how different people think through things. The questions are great. I mean, kudos to the moderators. I think we have a new set of moderators this year.
Yep.
But they all do such a fantastic job. And it's going to be really fun.
I think it'll be a lot of fun. So stay tuned. And as we know more and can share more about what we'll be doing in the session, we will not tell you what it is.
So, Alex, before we go forward, I have a question on the topic we're going to be talking about. So how do we pronounce it? Is it Lophomonas?
Bring in one of our guests.
Or is it
bring in a guest?
Lophomonas, or however you said it.
They're both sitting there with smiles on their face. Somebody set us straight. How do we pronounce it before we go on?
I always say Lophomonas.
Lophomonas, like loaf of bread.
Abhishek, how do you say it?
Yeah, me too. I say it Lophomonas, like trichomonas.
Okay, Lophomonas.
Lophomonas.
Like loaf of bread.
Loaf of bread.
All right. Well, we would usually re-record at this point, but because I'm on a tight timeline, we are just going to snowplow ahead.
Well, we're already learning things.
Well, let's meet our guests. We have a first-time guest, Dr. Abhishek Mewara. Dr. Mewara is an additional professor in the Department of Medical Parasitology at the Postgraduate Institute of Medical Education and Research in Chandigarh, India. Welcome, Abhishek. Thank you for joining us.
Thanks, Alex. Thanks for the invitation. It's an honor to be here.
And it's a pleasure to welcome back returning guest, Dr. Bobby Pritt. Dr. Pritt is the chair of the Division of Clinical Microbiology and professor of laboratory medicine and pathology at Mayo Clinic. Bobby, great to have you back.
It's a pleasure to be back.
The two of you, along with several co-authors, have a mini review in the January issue of JCM titled Lophomonas as a Respiratory Pathogen, Jumping the Gun, which kind of gives away the answer. But the title should, right? The title should have conclusions. But let's jump right in and talk about Lophomonas and whether it is a respiratory pathogen or not. And Bobby, we'll begin with you. Why did you decide to systematically review the literature about Lophomonas, and its potential role as a pathogen in humans?
Yeah, that's a great question, Alex. Abhishek, Blaine Matheson, Richard Bradbury, and I noted an increasing number of case reports of so-called Lophomonas as a human pathogen. But when each of us on our own had looked at the pictures in these reports, we weren't completely convinced. We actually thought they looked a lot like human ciliated epithelial cells. And so we were concerned that there was this large misidentification going on. We talked amongst ourselves and we had all come to this conclusion independently. So we decided to really do a full review of the literature, not come in with any conclusions, look at the molecular data, and really analyze for ourselves. Do we think that these are cases of human pathogens of Lophomonas, or is this misidentification? And to strengthen our study, we recruited Jillian Guile, who's a scientist who actually studies Lophomonas for her career, as well as Juan Zhao in Abhishek's lab, who helped with our extensive literature review, including Chinese language reports, which was a real strength of our ability to do the study.
That's awesome, Bobby. So, Abhishek, I am clearly not familiar with this organism, didn't even know how to say it correctly. So can you kind of, tell us a little bit more about the microorganism itself and, start from the basics? What are we talking about?
Yes, Ellie, I'm absolutely. I agree with you that even we didn't heard about Lophomonas until we saw all these cases of human beings reported with pulmonary infections of Lophomonas. So I kind of tend to agree with you on that. So basically, Lophomonas is a flagellate protozoan of the phylum Parabasalia, and the closest relatives of this flagellate are uh, symbionts, obligate symbionts of termites. So basically, it is found in the intestines of cockroaches. So there are various species of cockroaches where Lophomonas have been found. So these species are Blatta americana, the Blatta periplaneta, and several other species of Oriental and German cockroaches. And once we went into it, we found that there were two species, Lophomonas blatta and Lophomonas striata. So there's not much biology known about these parasites, but they were discovered in the late 1800th century, and they were discovered by Stein and Bushleigh. So there are only these two species known, and the closest relatives to which may cause human infections are Trichomonas vaginalis, and there are several other close relatives which are Tetratrichomonas, Pentatrichomonas, and Trichomonas tenax. So that is about the basic phylogenetic correlations of Lophomonas.
Thank you, Abhishek.
I didn't think I needed another reason to dislike cockroaches, but I think I found one.
That was a very helpful introduction. Much of your paper is a review and an in-depth analysis of the published literature about those putative infections with Lophomonas. What was your strategy for picking the papers that you included?
All right. So, since we decided to do a systematic review, we went through all the due diligence of selecting out the papers from various databases. So, for English language cases, we picked up Medline and Embase. And since there was a bulk of papers from China, we also included CNKI database, and we included Huan from to review these Chinese papers. So, after doing the title screening and abstract and full text screening, we came up with 100 studies. And of those 100 studies, we further sorted out as clinical case reports and case series. And then there were commentaries, some original studies, and then there were some reviews. So we sorted them out for four different kinds of analysis. So first we did a image analysis, all those studies which had images of any kind of images, be that microscopy or gel electrophoresis or PCR, or radiology pictures. And then we looked at studies which had videos, so for a video analysis. Then thirdly, we looked at studies with clinical data, clinical details along with diagnosis by microscopy or PCR. And then finally, we took some studies for a meta analysis. So we took 9 studies, and these studies had a large number of patients, and they didn't have individual data, but they had a prevalence kind of thing. So we wanted a pooled prevalence of the various reports of Lophomonas. So the strategy we had adopted, and finally we landed up with certain number of articles for our analysis.
Nice. Thanks, Abhishek. So, Bobby, kind of building on that, you guys looked at a number of case reports from Lophomonas infections, and you talk about how the identification of that is based on looking and finding the organism in respiratory samples like BAL fluid, for example, from patients with respiratory tract symptoms and infections. So can you talk to us a little bit about what the morphologic features of Lophomonas are that you would look for in those samples?
Yeah, absolutely. So I'll describe the morphologic features of true Lophomonas, Lophomonas bladderum, as identified in cockroaches, our favorite insect, as we just discussed. So true Lophomonas bladderum is actually very distinctive by light microscopy. It has this well-defined apical tuft of 50 or so flagella, and they all emerge at the cell apex, and they're tightly bundled together. So it actually gives the appearance of a horse's tail. That's what it's been described as. So it's not a carpet of flagella or cilia. It's in this little tight bundle. And then they have a single nucleus right below that tuft of flagella, right up in the apical aspect of the organism. So a very distinctive appearance.
So what non-pathogenic structures could be kind of confused with this organism, even though, as you just described it, it's pretty definitive what it looks like morphologically?
Yeah, I kind of already alluded to this. So Lophomonas bladderum could potentially be confused with human ciliated epithelial cells because they both have this furry-looking apical aspect. And most people have heard of ciliated epithelial cells. They're normal human host cells, and they line the human respiratory tract from the nasal sinuses down into the bronchi. They're also found in other places in the body, like the female fallopian tube, male reproductive tract. So it's not uncommon to see detached ciliated epithelial cells in human specimens like BAL fluid in urine, especially if there's inflammation that tends to cause the cells to exfoliate. Or if a brush is used to collect a specimen, like a bronchial brushing, it's very common to see detached epithelial cells. And these cells can be seen singly or in clumps that are called Crayola bodies. And sometimes just the tufts of cilia will detach. And it's a phenomenon that's called cytophoria, something that I learned when I did my cytopathology rotations as a resident in pathology. All of these can be mistaken as Lophomonas or vice versa because of that tuft of cilia. But thankfully, there's some reliable ways we can tell them apart. As I described, the morphology of Lophomonas is pretty distinctive. In comparison, the human epithelial cells that are ciliated have this distribution of cilia across the surface like a carpet. So it's not like a well-defined horse's tail. It goes all the way across to the surface. And also the human ciliated cells have a larger nucleus that tends to be in the more posterior aspect of the cell. So those are helpful features for differentiating them.
Got it. Thank you. So let's break the suspense. When you guys looked and reviewed all of the morphologic images that had been published and that you included in your analysis, what did you guys find?
Okay, yes. So we found 64 studies that met our criteria for inclusion. 49 were clearly human samples, 10 weren't directly related to human cases, and then 5 were from cockroach specimens. So we had a total of 79 images, and then they were placed into a spreadsheet that were separated from the article, and all of the authors went in and independently reviewed the images, and we categorized them as consistent with Lophomonas, not Lophomonas, or insufficient diagnostic features, low quality image. And our authors all come from different backgrounds. I think that's one of the strengths of our study. I'm a pathologist by training. Some of our other colleagues had extensive training in basic or clinical science, including Dr. Geil, as I said, who specializes in Lophomonas. So we did a consensus analysis, and after reviewing all the cases, we really didn't have any problems in coming to consensus. Sometimes it was between not Lophomonas and insufficient diagnostic features, but usually we could just easily come to consensus. So to come to the punchline, out of the 79 image sets, 42 were clearly not Lophomonas. 34 were low or insufficient quality, probably not Lophomonas, but just kind of fuzzy, out of focus pictures. 3 resembled true Lophomonas, and these were all from cockroaches. So none of the images from the human samples had characteristics of true Lophomonas. By morphology, we could say that there was no evidence of Lophomonas in any of these human samples.
Thank you, Bobby. That's very striking data. What about the videos? There were also some videos posted.
Yeah, I'm glad you asked,
to
Alex,
show it. Yeah.
because the videos are my favorite features. I didn't talk about the motility yet, but that's another really striking characteristic that allows us to tell Lophomonas apart from ciliated epithelial cells. So Lophomonas is a protozoan organism. It has this deliberate directional motility. And I encourage everyone to check out the videos we posted in the supplemental material. You can see the Lophomonas swimming through the environment with that anterior tuft of cilia. In comparison, the ciliated epithelial cells, they do have motility, which can be quite striking and even alarming when you look at them. You see the cilia beating, and they'll continue to beat as long as they have any residual ATP, but they don't go anywhere. They just sit there and beat. Sometimes they'll swim themselves around in a circle, but there's no directional motility. So even just looking at the motility alone is enough to differentiate by microscopy Lophomonas from ciliated epithelial cells.
And I agree. People really should go take a look at those videos. They're really striking.
Yeah.
And also the pictures that you included in the article are just fantastic. And even I could tell the difference between the ciliated epithelial cell and the supposed parasite. So check those out, everyone. Abhishek, let's turn to the clinical findings in the patients who were thought to have had Lophomonas infection. What did you find when you reviewed those papers?
So we had 38 studies which had clinical data, and these 38 studies had 58 patients. So when we looked at these studies, they were before 2015, they were mainly from China, but after 2015, the papers started coming from Iran, India, Peru, Ecuador, Egypt, and even one case from USA last year. So all these cases started coming up. And if you look at the age range, it's a very wide age range. There are patients of 3 months of age to patients 79 years old. If you look at the clinical features, so the clinical features range from upper respiratory tract infections to lower respiratory tract infections. So there are patients with allergic rhinitis or bronchial asthma-like features, and then there are pneumonia-like features. So there are both upper and lower respiratory tract infections. In pneumonia-like features, there was cough, hemoptysis, fever, all kinds of expectoration, and so basically it doesn't match. Either a pathogen will have upper respiratory tract infections or a lower respiratory tract infection, unless there are some viruses like COVID, which can have both URI as well as pneumonias. Generally, a pathogen like which causes pneumonia, let's say Mycobacterium tuberculosis, or a bacterial pneumonia like by Klebsiella or E. coli, they would cause pneumonia, but they will not cause URI. Whereas in case of Lophomonas, there are all kinds of infections and there are all kinds of samples. Surprisingly, Lophomonas has been isolated from sinuses, from nasal discharge, from sputum, from bronchoalveolar lavage, from biopsies, transbronchial biopsy, and also from urinary tract samples. So this is a wide range of sample and is not consistent with the pathogen coming from one particular organ system. So apart from these features, there were 90% of these patients had some kind of other comorbidity going on. So there were cancers of breast cancer, renal cell carcinoma, lung carcinoma. There were patients with diabetes mellitus, hypertension, COPD, pneumonias, tuberculosis. There were about 30% of these cases had an alternate microbiological diagnosis, which could explain the clinical features. Some had tuberculosis, some had bacterial pneumonias, cryptococcus, candida. So there were several other pathogens which were also detected. And then 60% of these patients had some kind of other antimicrobial therapy going on, and 90% of these patients were given metronidazole. So overall, it doesn't fit into one particular pattern. It's a very diverse kind of age range, male and female distribution, symptomatology, the kind of samples it is being retrieved from. So that was our consensus that the clinical features are very diverse.
Mm. Thanks, Abhishek. So, kind of along those lines, a question for you. How we never talked about how people would get infected with this organism, or what's the thought behind that? Is it a cockroach bite? Like, how are people getting infected theoretically?
Yeah. Yeah. So theoretically, or like I would say, it's more of a guesstimate. People just
Guesstimate.
guess that
Yeah.
it's it's the cyst of the Lophomonas in the from the cockroach's intestine, which is kind of floating out in the environment, and you inhale it. So
Mm.
by inhalation, you get infected. But there is no evidence for it. It is just speculation. And
Mm.
then when you say that, I would also ask that then, okay, what is the receptor? Where does it go and bind? So
Mm-hmm.
what tissue tropism it has? So all pathogens have some kind of tropism. So what tropism does Lophomonas have? So will it go to urinary tract as well as upper respiratory tract, and then also the sinuses and also the lungs? So that's kind of a very irregular kind of pattern for a
Yeah.
pathogen.
So we don't know.
There's no
Okay.
evidence.
No evidence at this point. Awesome. Thank you. So, one of the other things you guys did nicely in the paper is you included a small meta analysis of the prevalence of Lophomonas throughout some studies. So, can you talk to us about that meta analysis?
Yeah, so there were 9 studies which did not have individual clinical data, but there were a large number of patients. So we decided to do a small meta analysis for that. So for these 9 studies, 7 were from Iran, one from China, and one from India. And the cumulative number of patients screened was around 2,700. And out of these, 346 patients were reported to be to have a Lophomonas infection. So the pooled prevalence came out to be 17%. So that is a very high percentage of people infecting. But when we did an I-square analysis to look at the heterogeneity between these studies, we found an I-square value of 98%. So that shows a substantial heterogeneity, and that these studies are very diverse and they do not align with each other. And some of these studies had 1,000 patients, and only 1% of them were reported to have Lophomonas. The study from China had around 300 patients or so, and they had about 30% of them infected with Lophomonas reported.
Whoa.
So this was a very diverse and non-homogeneous data. This the not the heterogeneity of the meta-analysis could be because of the differences in the study design, the kind of population or the tests being done, but it could also be because there's no common factor in all those studies. The common pathogen is missing, therefore the results are so heterogeneous and diverse.
Hmm.
Thank you.
Interesting.
So we've talked about the morphological evidence or lack thereof. We've talked about the variability of the supposed clinical manifestations and the utility of that meta-analysis of prevalence. But we haven't yet talked about the molecular data. Bobby, several of the papers included molecular testing for Lophomonas using primers for the 18S RNA gene. What did you find when you analyzed the sequence of those primers?
Yeah, that's a really important question, Alex, because many cases were analyzed by this PCR that was first described in 2019, and it was used in Iran and then later in Thailand for diagnosing cases, these purported Lophomonas cases. And it was a bit troubling to us that many of the reports were based on PCR detection alone, and then they started omitting microscopic images. So it made it even more challenging to evaluate their claims. So we thought it would be very important to review the primers and the assay for its specificity and accuracy in actually detecting Lophomonas. We read the original paper of how it was evaluated and then described. And so the primers were originally designed against an alignment of a previously published supposed human case of Lophomonas bladderum, as well as the 18S RNA sequence of Lophomonas striata, which is a related cockroach commensal. And then importantly, the specificity of the primers were only confirmed by in silico analysis. So they didn't actually test any known Lophomonas controls from cockroaches, and they didn't do any true cross-reactivity testing with DNA from related protozoa. So we thought, well, let's delve into this a bit further to analyze the primers for specificity. So the good news is that our nucleotide blast search did show the primers have 100% sequence identity to Lophomonas bladderum and striata from cockroaches. So this assay could be used to reliably detect these two protozoa. But the bad news is that it will also detect other things. When we expanded our nucleotide blast of the primers against the entire NCBI database, we found that the primers also have 100% sequence identity and sequence cover, query cover to several other human-associated protozoa, such as Trichomonas vaginalis, Pentatrichomonas hominis, which is a commensal gut inhabitant, Tetratrichomonas species, some which are found in the human respiratory tract, Trichomonas tenax, which is found in the human oral cavity. So a lot of closely related species that also have 100% sequence identity. Therefore, if you're using this PCR, you couldn't call a positive as evidence for Lophomonas. You'd really need to sequence the amplicon to confirm the identity of the detected DNA, or better yet, develop a more specific PCR.
So the PCR assay is kind of acting like a serology assay.
Yes, we need our PRNT or our to confirm.
Would not be editors in conversation without you bringing it around to serology.
We got to come full circle. I won't ask about the availability of serology assays for Lophomonas.
Yeah,
seems like something that should be developed in a specialty laboratory, Ellie.
Perhaps.
Yeah, there you go.
But coming back to the PCR, so knowing that it's detecting other species and organisms, what did you guys ultimately find when you analyzed those sequences from Lophomonas detected in human samples?
Yeah, so these purported cases of Lophomonas bladderum, there were 18 sequences that had been deposited in GenBank. So we decided to analyze those sequences and see what they really were. And as expected, the top matches were themselves. They all match closely to one another, but they also closely matched up to 99% identity to Pentatrichomonas hominis and Tetratrichomonas species. They were not a great match for Trichomonas tenax, the one that lives in your oral cavity. So that's probably less likely the origin of these sequences. It's probably either Pentatrichomonas hominis, which is found in the human gut, or Tetratrichomonas hominis, or Tetratrichomonas species, which we know is found in the respiratory tract of humans. Interestingly, these purported species of these human Lophomonas bladderum sequences were only a 79 to 86% match to true Lophomonas that was detected from cockroaches. So I think that shows that it's not Lophomonas. It's one of these other trichomonads. So we can say with confidence that none of the published human Lophomonas bladderum cases are actually true Lophomonas bladderum.
Thank you, Bobby. Abhishek, let's pull this all together and put it in the context of Koch's postulates, which are, of course, the long-standing criteria by which we link a pathogen to a disease. And they are not perfect, but can you remind us what those criteria are and which of those criteria are fulfilled for Lophomonas as a cause of human disease?
Definitely, Alex. So Koch's postulates are what we teach in microbiology undergraduate curriculum to make students understand how a pathogen, a microorganism, is associated with a disease. So Robert Koch and Louis Pasteur had a hard time in their times to associate tuberculosis with pneumonias and to associate Vibrio cholerae with cholera. So they had to present in various scientific committees, and against a lot of criticism, they finally it took them a lot of time to convince. And therefore these postulates, they are kind of a background to understand how a pathogen can be attributed to the disease. So these generally speaking, if you say that a microorganism is causing a disease, that microorganism has to be present in abundance in that particular person, in that organ, and you should be able to isolate that particular microorganism. It should be culturable or some kind of identification, and then if you inoculate that same microorganism into another system, be it a tissue or an animal model or a human being, you should be able to reproduce that disease. And lastly, you could again be able to isolate that same microorganism from that second diseased system. So this kind of closes the loop and makes you understand that how a pathogen would be attributed to cause a disease. However, there are certain exceptions, like you said. So this is not true for the modern world. It was kind of a basic postulate in the 19th century, but now we know that there are organisms which can't be cultured. So you can't always isolate an organism like chlamydia, or maybe some non-culturable viruses or non-culturable bacteria. You can't isolate them, but you can then attribute the disease pathology. The other Cox postulates can be fulfilled. For example, a virus like HIV, for ethical reasons, you can't inoculate HIV in another human being and produce a disease to prove the Cox postulate. But you have other ways. The other Cox postulates are still valid. So it's kind of, you have to come to an understanding of how to attribute a pathogen to a disease. This due diligence has not been followed for Lophomonas. So first of all, when you identify it from the samples, the only way you identify is by microscopy or by PCR. The microscopy, as we all just now heard from Bobby, none of the microscopy pictures are the pictures of true Lophomonas. They're not convincing at all. The PCR again is faulty. It requires much more specificity to be developed. It is not validated. It is not tested against. No guidelines have been followed for the PCR. So that first Cox postulate doesn't hold true. Then there is little known about what tissue tropism, what pathology this parasite causes. It causes pneumonia. Has it been shown in histopathology to be there in the lung tissue? If we say that a parasite causes a disease, let's say entamoeba causes entamoeba colitis, then you will be able to show a trophozoite in the histopathology. So that way you can prove in some way by tissue pathology or by, and then there is a variety of organs. It is isolated. It is reported from urine, urinary tract. It is isolated from sinuses, nasal discharge, and several other kinds of specimen. So even if there are exceptions, there is still it has to make sense. And all this rigor has not been followed for Lophomonas, and therefore to us it doesn't follow the Cox postulates and not even the exceptions.
Mm-hmm.
Thank you, Abhishek. It's nice to have such a definitive conclusion from a paper. All evidence seems to point the same direction. Bobby, thank you so much for joining us. This has been a lot of fun.
Oh, it's such a pleasure to be back. Thanks for the invitation.
And Abhishek, thank you for joining us as well. I really enjoyed talking to you.
Yeah, my pleasure. Thank you so much for having me.
Mm-hmm.
Ellie, have you got an article picked out? Says the guy who is doing an episode after 2 fine AAC episodes because I was so slow to pick out a paper.
Well, I'm taking your lead. I'm a little slow. I haven't selected a paper yet, but it'll be exciting, I'm sure.
It will.
Whatever
I'm sure you'll pick something great.
Got
Great to see you.
Papers. You too. Thank you both.
Bye, everyone.
Thanks, everyone.
Bye-bye.
And thanks for listening.